Good evening, everyone. That was great. You got quiet on cue, so I'm pretty impressed by that. It's wonderful to see a great crowd of people out here tonight, and it's been an exciting week here on the Hill. We've had a homecoming with a few thousand of our alumni back on the Hill, and we dedicated a phenomenal new 12,000 square foot facility for our kinesiology program. Had great press coverage on that. And we had a huge visit from our partners from Shanghai today. And our colleague in biology, Dr. Katie Staub, has got a nice article on her in the Chronicle of Higher Education today, which in our business, that's like People magazine. So congratulations. If we haven't had a chance to meet, I'm Roger Casey, and it's my incredible honor to be the 9th president of McDaniel. And certainly my privilege and honor to introduce tonight's speaker as I welcome you to our first SmartTALK of the academic year. Through the SmartTALK programs, we highlight the McDaniel brand of liberal arts education, which is at work through the impressive achievements of our alumni. And many of these alumni have had an opportunity to connect to our Center for Experience and Opportunity, offering their career expertise, mentorship, networking, internships, and most importantly, jobs. So if you haven't had a chance to find out what the CEO does, I want you, after this event, to go to the back here and get a chance to meet Josh Ambrose and Aaron Benevento. I got to say it right because Aaron just got married, so congratulations, Aaron. And they're stationed right back at the back and can tell you about all the great opportunities you can have and the many ways that you can get involved through the CEO. And after this event, we're going to have a light reception right here in this room. So absolutely everybody's invited, cookies and punch, get a chance to mix and mingle and spend some more time talking to our guest tonight. Our guest tonight's Greg Merril. And Greg has been an entrepreneur from the time that he graduated in 1987, when he started applying his psychobiology degree, his creativity, and his uncanny ability to capitalize on trends in technology and in the life sciences with a series of successful business adventures. At the age of 22, he launched High Tech Plantations, which produced produced instructional videos for biotech companies. And he then partnered with his brother, who's now a physician, to launch his next innovation, a medical simulation program. Greg learned that 75% of the complications that arise from procedures like endoscopic biopsy occur in the first 30 cases of a doctor's career. So he decided to develop a surgical simulator with the goal of decreasing doctors' error. And High Tech Explanations became HT Medical Systems, which built surgical simulators and sold thousands of units. And in the year 2000, Greg guided the multi-million-dollar sale of this company. His next venture was Power Grid Fitness. It tackled obesity by developing an exercise device called the Kilowatt, which worked with any computer or console like an Xbox or PlayStation to combine the addictive entertainment of video games with a challenging isometric workout. At its debut in late 2004 at the annual Consumer Electronics Show in Las Vegas, the Kilowatt snagged the 2005 Best Innovation Award in the electronic game category. So the investment capital dried up in the 2008 economic downturn, and Greg turned his entrepreneurial enthusiasm to another program that kept going strong. And so we had the idea for Brain Sentry, while working with the military to develop a concussion assessment tool for the battlefield. And that company developed a sensor that attaches to the back of helmets, and it lights up when a player sustains excessive force and speed in a hit, and kept track of how many hard hits that a player had sustained over time. All of this was a goal to reduce second impact head trauma and brain injuries. And Greg's latest solution to a serious health problem was inspired by an alarming rise around the globe of multi-drug resistant pathogenic bacteria. Adaptive Phage Therapeutics develops therapies using targeted viruses, or phages, to kill so-called superbugs that are impervious to antibiotics. He's based in Gaithersburg, Maryland, and this new company is co-founded by Greg and his father, Dr. Carl Merril, who is a retired NIH researcher in collaboration with the United States Navy. And already this company's saving lives that you'll hear about tonight. So a remarkable career as an entrepreneur, as a life sciences innovator. Please join me in welcoming Greg Merril back to campus to share the story of his quest for what he calls the double bottom line. Help people and make money. Greg, great to see you here.
Thank you. It's great to be back on the Hill. So I've got it's a really fascinating story, this adaptive phage therapeutics. And the story starts with a vacation that Tom Patterson and his wife Stephanie took in November of 2015. They went to Egypt and they got on a river cruise down the Nile. They happened to both be infectious disease scientists at UCSD Medical Center, but that's at this point just a coincidence. On this river cruise, Tom Patterson started having really bad abdominal pains and he was throwing up, kind of like food poisoning. But it was so bad that they decided to call their doctor back home in San Diego. And the doctor said, don't mess around, this could be something serious, get to the local hospital. So the local hospital was in Luxor in Egypt, and they went there, and at the hospital they decided to do an upper GI endoscopy, where they put a fiber optic tube to look to see what's happening in the stomach. Unfortunately, the tube that they put in his stomach was contaminated with a bacteria called Acinetobacter baumannii, which happens to be the worst superbug bacteria in the world. So this is a bacteria that none of the antibiotics really work on. And Tom got extremely sick at that point. So sick that they had to put him on an airplane and fly him first to Frankfurt, Germany, where he spent 10 days and he slipped into a coma. And then they flew him back to San Diego, where he continued to be in a coma, and they hooked him up to ventilators and to IV lines. And his case, he was dying. And the month of December went by, and he's still in this coma. And then January, and he's wasting away, and his organs start to fail. And so they start planning his funeral, because nothing was working, and he was dying. And no one had ever survived an infection that had gotten that bad with this particular superbug infection. Now, luckily for Tom Patterson, his wife, Stephanie Strathty, is the Associate Dean of the UCSD Medical School. She has a PhD in infectious disease epidemiology, and she knew about something that happened and started happening on Earth 3.8 billion years ago, and that was life emerged on the Earth, and the life was the very simple life form of a bacteria, and co-evolving with those bacteria were little viruses that would kill the bacteria. And so this battle between these viruses and this bacteria has been going on for 3.8 billion years. And today, half of the bacteria on Earth today will be killed by these viruses. So the name of these viruses is bacteria phage. So phage is a Greek word meaning destroyer or eater. So these are viruses that kill bacteria. They don't kill anything else, they just will kill the bacteria. Actually, they were discovered on Earth 100 years ago by humans, and initially they were thought to be the cure for bacterial infections before antibiotics were even discovered. It wasn't until 1941 that penicillin was introduced, and that worked so well that's where the focus had been. So she knew about these bacteriophage. So she went to the doctor that was treating her husband, and she said, what about phage? Why don't we try phage? And the doctor said, look, nothing I'm doing is working. So if you could find a phage that would kill this Acinetobacter baumannii bacteria, I'll administer it. So she called the FDA, and she asked, do you know anyone who's working on these bacteriophage that would kill this particular infection? And the FDA knew about a research project going on by the US military. The Biological Defense Research Directorate of the US Navy had started working on a program that my father had started at NIH. And the program was a program where the use of phage to treat these kinds of infections. The idea with the Navy's program, they were very concerned about a terrorist using one of these bacteria to attack the United States. So they were really wanted to come up with a way of using the phage to address this threat. The military had another issue, which was at the time we were engaged in conflicts in the Middle East. And this particular multi-drug resistant bacteria was very prominent in Iraq and in the Middle East. So the military is very interested in coming up with a cure for this type of infection. She found out that the military was working on this program, and she called the military and said, "Could you let us use this experimental therapy on my husband?" And they agreed to try this. So in March of 2016, and at this point Tom had been in a coma for over, 4 months at that point, they shipped a culture of his bacteria from San Diego to Fort Detrick in Frederick, Maryland, where the Navy was doing this program. And what they had at Fort Detrick was they had collected sewage from around the world, poop, really. It's really a unique capability of the military to collect poop because they have bases all over the world. The Navy has ships, they go all over the world, and they were collecting hospital waste, which happens to be a great source for finding these types of viruses that will kill these types of pathogens that are resistant to bacteria. So they'd collected poop, they'd isolated from the poop the viruses that would kill the bacteria. Now, one of the challenges with these bacteriophage viruses is they're very picky eaters. So they have, in the scientific parlance, they would say they have a very narrow host range. They will only kill a particular strain of bacteria. So even within a species, say the Acinetobacter baumannii is the species, there are different strains within that species. And some of these viruses would kill a particular strain but not another. So what the Navy did was they went to the sewage and the poop, and they collected viruses, and they built up a collection of over 100 different strains of these phage that would kill the A. baumannii species, different strains of the species. And then they developed a robotic system that would simultaneously test hundreds of these phage against a patient's particular bacterial culture. They call that the host range quick test, or HRQT. And the collection of phage, they call the phage bank, like a library, a bank of these phage. So they shipped the culture from Tom Patterson to Fort Detrick, and they ran this process for the very first time they'd ever tried it on an actual human sample. And so they took 100 of these bacteriophage, they ran this test, and they found 5 out of the 100 would actually kill his particular strain of the bacteria. So they grew up the phage, which is done through a process of growing the bacteria, and then you put the phage in, and it kills all the bacteria, you end up with a tube filled with viruses and bacterial guts. And then they had to get rid of all the bacterial guts because that can actually make you sick by itself. So they had to grow the phage, purify the phage, and they, on March 11th, 2000, this was actually March 17th, 2016, they delivered those 5 purified phage to San Diego to inject into Tom Patterson's IV line. And when they injected the phage into his IV line, within 48 hours, he was awake. It was a miraculous recovery. No one had ever survived that type of illness. The Navy and everyone involved was just ecstatic, as you can imagine. They said, well, maybe this was a fluke. Let's try this again. So they found at Children's Hospital in Washington, D.C., a 2-year-old child that was dying from a heart infection, a multi-drug resistant Pseudomonas, which is a different species of bacteria. And they tried it on this 2-year-old child. Sure enough, the infection went away. And at that point, they thought, well, this works. We need to get this to market. We need to somehow figure out how to get this through the FDA regulatory process. So they asked the pharmaceutical industry, the different big companies, if they would help bring this to market. And the pharmaceutical companies really didn't know what to make of it. It was very different from the types of products that they had brought to market in the past, the more typical drugs that you can put in a bottle and put on the shelf. This was a process in which you'd have to get the sample, you had to do this thing, you had to bring something back. So the pharmaceutical company said no. So the Navy, it had actually involved my father in the treatment of Tom Patterson because they were going to try it on a human for the first time, so they wanted to know, well, what's the appropriate dose? How should we administer this? So my father helped out with that. So when it came time for the Navy to try to figure out how to commercialize this, they asked him, could you help us bring this to market? My father's initial reaction was, I'm a scientist, I don't know how to do that. But he told me about it, and I thought, this is the biggest thing that any life science entrepreneur could ever get their hands on. And this threat of multi-drug resistant bacteria is becoming the biggest threat facing humanity right now. The World Health Organization says that by 2050, if no one figures out a way to treat these bacterial infections, these infections will kill more people than any other type of illness, more than cancer. 10 million people a year will die from these types of infections. It'll be just like before penicillin was invented. So I thought, well, this is an amazing opportunity. If we could get the Navy to really agree to help us. So I put together a proposal to the Navy. I said, look, my father and I will do this. We'll help bring this to market. We'll take this through the regulatory process. If you can agree to give us the worldwide exclusive commercial rights to your collection of viruses and agree to continue to add viruses to that collection, and we want the worldwide exclusive rights to that technology to match these viruses to a patient sample, and help us with the clinical trials and any research that's needed. And the Navy said yes. And the reason they said yes is because getting this cure to market is critical to their mission. They needed someone to do it. The big pharmas weren't stepping up, so it was a real amazing opportunity. So when they said yes, then it's like, oh boy, now we got to figure out actually how to do this. And there were some challenges. One of the challenges was related to how do you make this commercially scalable? So the Tom Patterson case was really interesting. Of course, that was the model for this business. But the Tom Patterson case involved a microbiologist at Fort Detrick getting this bacterial sample and not sleeping for an entire week while he worked on getting this phage grown and purified and delivered. You'd have to have all these insomniacs working in your lab to make that commercially viable, and that's I guess not ethical. But so we looked at that process and thought, look, how could you make that scalable? And one of the things we realized was that this phage bank, this collection of viruses, it's big, but it's not infinite. What if we were to pre-manufacture all the phage? So we're going to grow all the phage individually, we're going to purify all the phage, we'll put them each in little single-dose vials, and we'll put those in inventory. So all that time that was spent growing and purifying, that's going to be out of the critical path of treating the patient. So now we took a process that took a week, and we reduced it to just the time it takes to run that test to see which phage will match the patient sample. Now, when the Navy first started working on it, that test took 24 hours. We got it down to 8 hours. So now we can commercialize a process that takes 8 hours to run the test, and then whatever logistics time to get the samples and return the sample to the patient. So that was a problem that we had to address. And you just figure out conceptually that was what we could do. Then you have to figure out actually how you're going to do that, how you're going to build the manufacturing facilities, because it turns out that in drug manufacturing, producing viruses like that is not a typical process. So we had to build our own manufacturing processes and our own manufacturing facility just to support clinical trials for that. So that was definitely one of the challenges that we've been faced with. The other challenge was the FDA. So the FDA didn't know what to do with this. I mean, my first question when we were awarded this worldwide exclusive agreement with the Navy was, okay, so what is the product? I mean, there's this collection of viruses, the FDA approved drugs. What is the actual drug? Is it each individual virus? Because if it's each individual virus, then each virus is going to require a clinical trial. If you have 1,000 of them, it's going to take like 400 years to do the clinical trial, and that's going to be really expensive, and I'm not going to live long enough to see this thing actually work. So that wasn't going to work. But fortunately, the FDA was really motivated to try to help us out because they didn't have any drugs that would work, so people were dying from this. We also had the advantage that the phage, these viruses, are known to be safe. In fact, we've all been injected with these viruses unintentionally. It turns out that a lot of the vaccines are actually contaminated with these phage, and they're totally harmless to humans. Unless you're a bacteria, they really aren't going to hurt you. They only will infect and kill a bacteria. It's a byproduct of the manufacturing of vaccines. And even cancer doctors will inject these phage into patients just to test the immune response when the patients are coming off immune suppressants. So the FDA said, okay, look, we know this is safe. What if we were to just define what specific safety issues there might be for all of these phage, and we look at the phage. They wanted us to sequence them, for example, and then look for any genes that might be in any of these viruses that could be dangerous, like a gene that might transmit antibiotic resistance or might transmit some production of some toxin in the body. So those particular genes happen to be known. You can actually go online, you can find the sequences for those bad genes. So we sequence the viruses, we look for those bad genes, and then we produce all that information and present it to the FDA, and they can approve this entire collection without actually putting each individual virus in a human. And they agreed to that, and so that really makes this business viable. So what we're doing now is we're continuing to treat patients on an emergency basis. So in order to get this treatment right now, because it's not FDA approved yet, you have to have failed standard of care therapy and be either about to die or about to lose a limb. And we've had continuing amazing success in treating these patients. The FDA is encouraging us to save as many people as we can. So that's what we're doing. In the meantime, we've built a manufacturing facility in Gaithersburg, Maryland, that's big enough to support our clinical trials. And we're now putting together materials to start doing the work to get broad approval by the FDA to go to market with this therapy. And an interesting thing about what we have is that this platform is very broad in terms of the kinds of infections, type of bacteria we can treat. So once we get going, we're really going to expand, and it's going to become useful for a lot of different types of infections. So we're working on that, and we are also looking forward to implementing another technology, which is artificial intelligence. So one of the things that's critical in this type of infectious disease treatment is to get the treatment to the patient fast. So a lot of times these patients are, when they become septic, that is, the bacteria has gotten into the bloodstream, it's 50% of the patients who get to that stage will actually die. So you have to very quickly get the therapy. And so in our process where we have to match the patient's bacteria to this phage collection, that could take, even at 8 hours, that might be 8 hours too long. So right now we're working on sequencing bacteria that we collect from hospitals and taking those sequenced bacteria and then our phage bank, which is all sequenced, and running them in the laboratory and building a data set and teaching a computer to start to predict which phage will kill which bacteria. And over thousands and thousands of tests, the machine will learn to, when you submit a new bacterial sequence, to know which of the viruses will kill that bacteria. At that point, it'll be instant in terms of knowing which therapy to work, just as fast as pulling a bottle of penicillin off the shelf. And so that's a big part of our vision, is that that's what's going to happen. And at that point, we will own the infectious disease market. Because when you go to the hospital in the future, they'll be doing sequencing to diagnose which bacteria you have. And if you know the sequence information, we'll be able to tell you exactly which therapy will work to cure your infection. So we'll control that decision-making process. And that's our vision for this process moving forward. Thank you.
So you guys have come up with a lot of good questions. So I'm looking forward to this conversation. And let me start with this question, Greg, because I think everybody heard from the introduction all of the different companies and ideas and things you've been involved in your life. So when someone says to you, so what do you do for a living? What's your career? What's your one sentence or less answer to that question?
I think that I usually identify myself as an inventor. So more than an entrepreneur, which is a little bit hard to kind of put your head around, what does that really mean? I think of myself more as an inventor.
And when did you know this is what I want to do in life?
So I entered my entrepreneurial career out of fear. So I didn't like school. I struggled with school. Now I'm convinced that I'm dyslexic, but I was never diagnosed as dyslexic. But I really had a hard time with formal education. And I got a degree here at McDaniel College in psychobiology, but I did not want to go to grad school. My father would always joke that I could do anything I want with my life after medical school. And fortunately, my brother went to medical school. He's 3 years older, so that kind of took the pressure off me. But because I didn't like school, I didn't have great grades, I felt like graduate school wasn't in the cards. I didn't want to go to graduate school, so I sort of took that off the shelf, off the possibilities. And then the idea of getting a job was, I was a little bit embarrassed about my grades. So I didn't want to have to get a job. I couldn't even think about what kind of job I would get. So I remember after final exam senior year trying to figure out what am I going to do. And I had always been interested in the arts. I took a video class here. I produced videos in high school. I won the film festivals in our county, and I don't remember if I won the state festival, but I was really interested in video. So I thought, why don't I take my interest in video and my background in science and combine those, and that was the origin of my first company, to explain technical things using the arts, using videotape. And I simply called up different scientific equipment companies and asked them, do you need for people to understand how to use your equipment? How about if I produce a video for you? And I landed my first contract, and that led to the next contract and the next contract, and pretty soon I was producing films for big companies like DuPont and Bio-Rad Labs, and built up a business that way.
So you didn't have great grades. Where are you getting the affirmation that you actually know what you're doing? I mean, how do you get the confidence that I really do? I have the skill set.
I'm trying to answer that myself. I think there might be an advantage in always wondering if you know what you're doing, sort of always questioning yourself. I think I'm not very confident in what I'm doing, but because of that, I'll do the research to figure out, maybe I'm not so, I just assume that I don't know what I'm doing, so I have to look it up. I've got to ask people who do things and listen to them and see, does that make sense? And let me ask somebody else and sort of collect data points and then sort of figure it out and go a different way. So that's what I do.
So it sounds like you were actually pretty successful right off the bat, but when you talk to entrepreneurs, you always hear failure. You've got to be comfortable with failure. So what was your first big failure?
So you in the introduction, you talked about these different companies, and really being an entrepreneur, at least in my experience, it is totally a roller coaster. I mean, even within the success, there's ups and there's downs, and the first company ended up being a big success, and a lot of people made money off my first company. I think we had a $1 billion market cap at the peak of the NASDAQ. But the next company I thought was a great idea, which was the exercise equipment for children when they play video games, plugs into the Xbox. And we got up to a couple million in revenue very quickly with that company. But then the economy collapsed in 2008. Just as we had done a deal with Microsoft and with Nintendo to introduce the technology for their next generation game controllers, and we were out raising capital to bring that to market, the economy collapsed, all the money evaporated, and that ended up being a down deal. I mean, I think I lost several $1 million of my own capital trying to get that going. I was able to we pivoted and used the technology for some military training and ended up licensing the technology to Nintendo for their game systems. But that was probably, I would say, the first failure of many.
So how do you pick yourself up after that?
I think it's fear. I think it's, then your back's against the wall and it's like, what am I going to do now? What are the options? I feel like I'm not employable. I don't think anyone would hire me. So I'm like stuck. I have to sort of think of stuff and go out there and do it myself. That is my attitude. I have had a relatively normal job. After the exercise equipment company evaporated, I took a job as a chief operating officer of a defense contractor that made missiles and radar systems, and I hated it. It was, going there 9 to 5. One of the things I remember driving to work, and in defense contracting, a lot of it is you find an engineer, you put them in an office to do missile design or whatever it is. And I realized that if I lost my job, if the company went out of business that I'm working for, nothing in the world would change. The engineer that's working on that missile is still going to work on that missile. The only thing that's going to change is that the logo on his paycheck will be different. Mm-hmm. And so that was just destroying my soul. So I can't do that. So I have to just think of my own stuff and do it that way.
So you've been described too as a serial entrepreneur. Do you sort of wake up in the morning and here's an idea and you toss that out and let it go down the shower drain? Or do you find yourself really sort of obsessed by a single thing and then you pursue that out to its fullest? Or none of those?
So there's definitely, now I've gotten to the point where there's definitely criteria that are used to evaluate ideas for pursuing as an entrepreneur. I mean, there has to be some intellectual property or some way of protecting the concept going forward. So I'll typically look at what the intellectual property landscape looks like. That means do some quick searches on the patent office to see, is this something that's defensible? I mean, that's really related to technology types of companies. But then you also want to know, is it a need? I only want to do things that there's a need for because the need translates to a market. And there's, a lot of people will say, this is a really cool idea, and it might be a really cool idea, but if no one needs it, then it's just a cool idea. So I go through this vetting process in my mind. I mean, is there a market? Is it an unmet market need? Can it be differentiated from what's out there in terms of both intellectual property and maybe some other attributes? So I mean, I think I go through this process, and it's not easy, and you have to vet a lot of ideas before you get to the ones that because being an entrepreneur is an all-consuming. It's not 9 to 5. It's you waking up in the middle of the night with an idea and write it on a notepad by the side of your bed. You can't be willy-nilly with taking on too many of these ideas because it totally consumes you 100%.
When you first gathered the ideas for adaptive phase technologies and therapeutics, and you heard the idea, did it pass the vet test, or did you have to really think a long time about this?
There were some questions. The need, clearly there. Unmet need. The antibiotics aren't working anymore. Everyone agrees with that as a problem. The phage seems to work, so that's great. The intellectual property, the patents, were a question. Is this protectable? So that was a question. Is it scalable? So these questions that I talked about in my little talk about, it's got to be scalable. We can't have microbiologists, millions of them not sleeping, trying to cure all the people in the world. So that was a question. So these were questions. So I would say that on its surface seemed very promising enough to do a deep dive, and it took me a couple months before I got my head around it and made the proposal to the Navy to take this on.
You've walked away from businesses. Businesses have failed. You've started new businesses. How do you know when the time is right to leave?
That's a tough question. Usually, I know it's time to leave about a year after it was actually time to leave. I think it's part of the entrepreneurial mindset. It's always going to be ups and downs. You just fight and you fight and you fight, and you try something else. I sometimes compare it to like you're in an airplane that's crashing, and you've got all these controls in front of you. So you start pulling all these levers, and there's always another lever to pull. And so you're never quite sure, why is it failing? Maybe if I did this, maybe if I did that. And so that's a really good question, and I don't know the answer.
Can you give a good example of when you left something at exactly the right time?
No.
Okay.
but here's a really interesting example. So I started this company called Brain Sentry, and the company was trying to address this problem of mostly kids who play sports that involve head impacts. And the problem is obviously concussions, but it's also the long-term effects of hitting your head, the chronic traumatic encephalopathy. It's multiple head hits. Even if you don't get a concussion, it's not good. Basically, don't hit your head. So I thought that's a great idea. Let's put the sensors on the helmets, and we'll be able to monitor head impacts. I remember originally, so I was vetting this idea. Like I was going through this vetting process. And one of the things I did in this vetting process was I took a youth football coach out for a beer, and I told him about this idea. I said, "What do you think?" He's like, "It's a great idea." I said, "Great. Would you use it on your team?" He's like, there's no effing way I would use it. What if it's 3rd down and 2 yards to go, my star player has his light going on his helmet? I'm like, well, you take him out. He said, I'm not taking that kid out. This guy was coaching 10-year-olds in tackle football. And so that should have been my warning sign. But of course, 2 years later, millions of dollars invested in trying to get this to market. And we had every player in arena football was wearing my sensors. We had a couple youth leagues using the sensors. But the youth football is, one of the oversight organizations is a group called USA Football. They're funded by the NFL and the NFL Players Association. And one of the big programs they have is to train parents in how to be football coaches. So they have a program called Heads Up Tackling. And the idea is to get the parents to teach the kids to tackle without hitting their heads. Now, I became a Heads Up Tackle football coach through their program, got certified and all that. I went to the director of USA Football. I said, if we use these sensors, we can monitor whether the parents are doing a good job teaching the kids. They should start off with some number of head impacts in their practices, and by the end of the season, they should have a smaller number. And they said, well, there's really no science to say that would work. So I said, okay, well, why don't we work together? We'll figure out what science needs to be done to decide whether this will work or it won't work, and I'll pay for the research. And he said, we're not interested. So that's when I started to realize that one criteria of the market. So that was an example, again, I think of a good idea, a need, but the market wasn't there. They weren't ready for that solution.
When I visited with you the very first time, I remember coming into Brain Century and you shared the story of, and pictures of, here's somebody who died because of this. And then when you started Adaptive Phage, here's the story of a guy who almost died. You really seem to be obsessed with this notion of at the heart of the entrepreneurial choice is helping people. Mm-hmm. There are a lot of entrepreneurs, the very first conversation I have with them is always about money. Can you talk a little bit about, are you unusual in this regard? Are you?
So I don't know if I'm so unusual, but I do, for me, it's not just about the money. It is about these two bottom lines. The bottom line being, I want to feel like I'm doing something that's meaningful, because it is all-consuming. This is your life. So I want to feel like if I'm successful, it's going to benefit society. I mean, that just, for me, is really important. And then you want to be able to make money, in part because I guess it's nice to make money, but it's also a way of sustaining that and accelerating that social good. So I think there's why it's definitely not unique to me. I mean, the people talk about social entrepreneurship, and I think it's really a category of entrepreneurship. But for me, that's always been a driving force.
So let's spend some time on adaptive phage here. So what do you find so interesting about viruses?
Viruses are amazing. These phage, if I wish I had a picture to show you, but you could go to our website at aphage.com. You'll see pictures of these things. They look like lunar landing ships. I mean, it's hard to believe that nature came up with this design of this geometric head thing that's made out of protein, and it has DNA inside it, and it's got this stalk and these legs, and it lands on the bacteria like a lunar lander, and then it injects the DNA into the bacteria. And then it takes over the bacteria and turns the bacteria into a factory to make more viruses. It's just crazy that this stuff is happening. And the other thing that's crazy about it is that we are just at the beginning of understanding how these viruses affect life on Earth. And so it's neat to be participating in this learning that we're all going through on what viruses are.
So what happens to this virus after it starts multiplying, destroying things? How does it get out of your body, or is it always in the body?
No, it's a good question. So the viruses, the body looks at the viruses just as little proteins, and the liver will filter out these viruses very quickly. In fact, that was one of the challenges in using this phage therapy the way we do. We inject it into the patient, and the patient's the circulatory system, the liver will filter out most of these viruses, treats it just as food, so you just sort of digest it. Some of the viruses will end up in the spleen, which is a really interesting point by itself, because it turns out that when these viruses end up in the spleen, they can sit there forever. And they're totally viable. They're just these little microscopic proteins with DNA, and they're just sitting there. And it's so weird that they sit there that we think that it's probably on purpose, that we've evolved to store these phage, because if you think about it, if the patient who has these, or any of us, when we're exposed to another bacteria that phage may kill, when that bacteria is in our bloodstream, that phage will become active again, and it will replicate, and so it becomes part of our immune system. No one's ever even published that, but that's what I think is happening. And that's why this is such a new and exciting area, is that we're learning about how we live. I guess, now we talk about people in biology class, you learn that humans are made up of, our cells are defined by the DNA in the cells. But then they say, well, there's probably actually more DNA in us that's from the bacteria in our guts. If you look at all the bacteria that's and all the DNA in the bacteria in our gut, we're probably more of that than we are of the human genetic material. And then there's all these viruses. So we're very complicated, more complicated than I ever learned in school. And that's neat.
You said several times in your talk, the FDA said, well, we know this is safe. We know this is safe. That sentence is usually at the beginning of every zombie movie.
Yeah.
Is there no sort of Frankensteinian type possibility here in the kind of work that you're doing?
Yeah, there is. The phage are probably responsible for one of the biggest epidemics on diphtheria is probably transmitted through a phage. It's transmitted through a phage. So phage can be dangerous, but we know the toxic genes, and so we can screen for those. The FDA believes phage are safe because we've all been injected with phage unintentionally, and phage really generally is safe, and so we know what to look for. And so for us, the FDA, they're not just saying carte blanche, oh, phage are safe. They say, look, we want to screen the phage you have for certain properties that are known to be potentially dangerous. Let's not use those, and let's use the ones that don't have those properties.
You saved every patient you've been able to get to so far, right?
There was one tragic patient who we didn't get to on time, and you may have read about her in Time magazine. She was 25-year-old Stanford grad, two-sport athlete, cystic fibrosis patient. She had a lung transplant back in September of 2017, and her new lungs had become infected with a Burkholderia bacteria, which was resistant to all the antibiotics. And so she was going downhill, and she was admitted to Pittsburgh Hospital. And her parents decided they would try the phage, but by the time that they asked for help, their daughter the disease had really progressed. She went into a coma and started to have organ failure. We chartered a jet. We produced the phage. We flew the jet from our labs to Pittsburgh. Had a helicopter fly it to the hospital. Her parents were live streaming the helicopter landing on Facebook, and they got a dose in her at like 2 a.m, and then she passed away the next day. So it was just too late. But otherwise, the phage has been. We've cured quite a few patients that traditional therapies had failed.
What's the most recent success story?
Well, the one that I told you about, which I think is very dramatic, which was like 3 patients ago, but I think it's a good story. It was a patient in Israel. So there was a guy as a taxi cab driver, and he was in a car accident, and his leg got all torn up, and he had this big gash in his left leg, and it became infected with that Acinetobacter baumannii, the same infection that Tom Patterson had. And he also had a multi-drug resistant strain of Klebsiella, and his leg was festering. It was disgusting. They took a flap of muscle to try to get the wound to heal, and it just wasn't healing. So they scheduled him to have his leg amputated. They reached out to us and they said, maybe we can get the leg to just survive a little longer. If you could, maybe you could help us and find some phage. So they sent us the cultures, and we found the phage. We sent them back, and they injected the phage into this guy, and within 2 weeks the wound healed, and he was out of the hospital with his leg. So that was, an example of one of these miraculous recoveries.
Who's the competition? Is there?
So there are a number of companies that are working on phage. We are the only company doing this kind of precision-matched approach, and we think we're the only company that's going to be successful with phage therapy, because the other alternative would be to take some of these phage and put them into a cocktail, a mixture, and just have them on the shelf, so when someone gets sick, you can just administer the phage, and maybe it'll work. I say maybe it'll work because you don't know if it's going to match the particular strain of the bacteria. But it's saying that you had enough phage in there and it did match, you could potentially save people. But we earlier talked about the narrow host range of these phage. They're very specific. And one of the drawbacks of being very specific is that the bacteria, when it mutates just a little bit, can work around the viruses. So the resistance that we see with antibiotics, you'll see that same resistance with a phage mixture that isn't, custom matched to the bacteria. So resistance is a problem. And in fact, the Tom Patterson case, I didn't tell you another part of the story. So he had that miraculous recovery. 2 weeks later, he started getting sick again. And they did another culture of his infection. And he still had Abumani, but now it was a different strain. So we ran the process again. And now we found 2 different phage. And those 2 phage, in combination with his antibiotics, was able to save his life. That's why we called the company Adaptive Phage Therapeutics, because we can adapt the therapy to the patient's needs and to the disease as it's progressing. And that's, no one else is doing anything like that.
So why wouldn't Big Pharma be interested in this?
Well, so Big Pharma is interested now, but initially they weren't. Initially, they said, we've done this before, it doesn't work. In the 1920s and the 1930s, the Big Pharma, they were working on phage. Lilly was working on phage. Before Fleming discovered penicillin, this is what they thought would be the cure for bacterial infections. But the problem was sometimes it would work, and sometimes it wouldn't work, and that was because they didn't understand it at the time. But this narrow host range of the phage means that you can come up with a mixture that will work for somebody, and you try it on another person, and it doesn't work, because they might have a different strain of the infection. So the phage as a therapy has a bad reputation in the pharmaceutical industry. And so they thought, well, this isn't going to work. And then of course, they start seeing in Time magazine and in the news and that we've cured one person, then we cured another person, then we cured another person, then we cured another person. And so now 3 of the top 10 largest pharmaceutical companies are in discussions with us for partnerships.
How big is the superbug problem now, and how big is it anticipated to be?
So in the United States, the CDC says that there are 2 million of these superbug infections right now. 35,000 people will die from these infections, which is still a relatively small number, but the fear is that they could be a major outbreak with one of these types of bacteria that has this capability to resist antibiotics, and then we'll be in big trouble. And in certain parts of the world, you see more and more of these drug-resistant infections. So it's becoming a bigger and bigger problem. And as I mentioned at the beginning, the World Health Organization has identified this as the biggest up-and-coming threat to humanity.
So you've invented or refined some real new technologies in the work you're doing. What's the next step so that someone in Seattle has a problem? How are you going to get it to them in time? I mean, what's the strategy for that?
So I mentioned this a little bit in the talk, but this is a big part of the challenge. We need to make it so that take the logistics challenge of collecting samples and delivering phage out of the equation. And so the way we do that is we're going to have a dispensing system that goes in the pharmacy, the hospitals. And that dispensing system is going to contain thousands of little vials of a wide variety of different strains of the phage. So that'll be right there in the hospital. And then the next challenge will be, well, how do you do the testing? So initially, we're going to be putting our testing system in laboratories just like if you have to take a urine sample to a lab like a Quest Diagnostics or a LabCorp type of facility, well, those facilities in the future will be running our test to test for which of the phage will work. But then in the more distant vision, we'll be using artificial intelligence and computer software to instantly match. So that'll happen right at the bedside, and then the therapy will be available just as fast as you could take a medicine off the shelf. You can take a custom matched, precision matched therapy that is demonstrated to have be effective in treating that patient.
So your dad said, after med school, you can do anything you want, and obviously that didn't happen. But here you are now, you're working with your dad on something that was critically important to him decades ago. What's this like, and how do you feel?
Well, it's really neat. This phage stuff, I didn't really have a grasp for what it was until the last couple of years. But it was like the soundtrack of my youth. My dad would talk about these phage. I don't know what the heck he was talking about. He's doing basic research on this stuff, and he'd talk about how, we have this mouse, and we gave him some bacteria, and it was on its back with its paws in the air, and then we gave it this phage stuff, and he jumped up and he was running around. I just, okay, Dad, whatever. To get a different view of my father at this stage, he's in his 80s, and he's retired from NIH. Now I sort of pulled him out of retirement to help us with this. But I have a totally new appreciation for what he did. And for him, he gets really emotional about this because he spent his whole career at NIH doing this kind of basic research, never really directly seeing the effect of his work on patients. And now we're seeing these patients, and they are, they're alive and they're coming back. Tom Patterson and his wife, Stephanie Strathley, are now investors in our company, and they're on our advisory board. And so we have a close relationship with them, and they come into town, they have dinner with us, and it's really neat.
So what's next for the company? You need to expand, you need to build, you need to raise more capital. Tell us a little about this.
So, in the sort of the business evolution is pretty well laid out in terms of you have to go through this regulatory process in order to get an allowance from the FDA to go to big market. So what that means is you have to do clinical studies. So we built a facility that's big enough to support the production of these phage materials in enough quantity to support the clinical trials. But to be at a commercial size, we need to have a huge facility. So I don't know how many square feet Decker Center is, but we're talking about, I don't know, but the facility that we're talking about building now is 150,000 square feet, which is quite a large facility. With hundreds of workers producing phage and purifying phage. And the automation is really, I love the toys, the automation. One of the things that we're going to have in this new facility is huge freezers that are at negative 80 degrees, the temperature of dry ice. And these freezers have robots inside, and they can place the vials. And so you can on a keyboard say, well, this patient needs these 4 phage, and the robot goes in the freezer and picks out the phage and puts it into a package for you. And this freezer is like the size of this room. So we'll have those in this new facility. We have other really innovative robotic systems for doing the manufacturing that no one else has ever had to do before. So we're trying to pushing the envelope in the drug manufacturing area, and then in these areas of artificial intelligence, and of course the use of the viruses. So it's all sort of implementing that vision of scale is what we're working on.
So I know you're going to privilege hiring McDaniel alumni. So what's your advice out here for people who might want to work in your business here in the next decade as they finish school or go to graduate school?
Well, yeah, I think what we're doing is very technical, and so gaining the skills in microbiology is for the employees that we have right now are almost all microbiologists. Most are actually just at the college level. They finished college courses in biology, had experience during college in working with phage. They all had that, so that's really important. But so I would say for us the microbiology, and as we expand and we have more diverse requirements in terms of business development, there's of course the areas for business development, but what we're doing is very cross-disciplinary. So it's not just business, it's a business combined with pharmaceuticals, or microbiology combined with manufacturing. So I would say that learning some of those things would be helpful.
Where did you learn cross-disciplinarity?
The School of Hard Knocks, and also, I think that's one of the advantages of a liberal arts education. I really think that if you were to go to a school that's more of a focus in a particular discipline, you wouldn't gain that more broad perspective that comes from a liberal arts education. So I think you get a great start at a school like McDaniel in terms of having that broader exposure to the liberal arts, and including the sciences. So I do think that this is a great way to get started with that broader perspective.
When you look back at your undergraduate experience here, is there a particular course or set of experiences that you had that now you think they really were formative in terms of getting you to where you are?
There are. I mean, some are totally non-intuitive. One, this is kind of somewhat of the entrepreneurial thing. When I came here, I played the violin, and there wasn't a string music program here. So I went to the chairman of the music department, and I said, I play the violin. He says, oh, well, I play the viola. Why don't you come in, we'll start a class, and we'll just play duets. So I started playing duets with a music professor, and then we brought in a couple other people. By the time I left, we had an entire string ensemble, including the community. And so that experience of building this organization was non-intuitive, maybe non-traditional learning, but that's the kind of thing you can do at a school like this, is you can do things like that. So that was definitely one of the experiences. And of course, at the time, I think I was one of the first graduates in this new degree called psychobiology, which was a combination of psychology and biology. And so, that even the fact that it was a new thing, I wasn't limited by a fixed things. Here we could do, you could do anything you wanted.
So if we have a student here who's struggling in a sophomore bio class, the grades aren't that great, what's your advice for them?
Keep plugging away. It's not the end of the world. Don't give up, even if you do end up with bad grades. It does limit what you can do, and so you'll feel like your back's against the wall. But sometimes the best things come out of feeling like your back's against the wall.
You've done an enormous number of things in your life. How do you think this is the last one that you want to build out, or do you have any idea? When you were in the middle of your last thing, did you think this is the one I'm going to spend the rest of my life doing?
I mean, you hope that this is going to be so big that okay, I'm done. No, but I think that I'm too driven to. And the other thing that's kind of interesting is when you've done this a few times, and then people start coming to you with their ideas and they want help. So sometimes you can really get sucked into other people's ideas. And so I imagine that, assuming that this continues to go in the trajectory that it is, and it is very successful, I'll probably become more of a mentor and probably just an investor in startup companies and try to add my whatever value I can to other people's ideas.
Is this the thing you've done that you look back and you go, God, this is what I'm most proud of, or not?
But the first company was, was a big, was interesting because I really didn't know what the heck I was doing in terms of business. I didn't take any business classes here, so that was really just being terrified of the whole business concept. I remember, and because of that, being so, I was so uncomfortable with my knowledge of business that I really dove in, and I started going to all these community courses in business, and I hired an accountant not to do my books, but every week I'd go in and he'd teach me how to make ledgers and do the books. So I was starting to learn about the financial parts of it. So the first company was clearly something that I really cherish, that adventure. This one has the promise to being bigger than anything I've ever been involved with. So, I'm really eager to see where this goes.
Well, we're very eager to see where it goes. We're incredibly proud of what you've achieved as a graduate of this college. Students who are in our entrepreneurship program know Greg because he comes back and works with them and gives his time and helps out. And I see a lot of future employees. I just, their faces out there. So we're honored that you came back tonight. I know the most important thing that I think that's in your office is a Western McDaniel degree, but I also want to make sure your office has a McDaniel degree hanging in it. So,
that's awesome.
Would you please join me in welcoming our McDaniel alum, Greg Merril, tonight, and thank you for being here.
Thank you. That's great. Thank you.
I want to thank you all for being here. November the 13th is our next Smart Talk. Jim Wilberger, who's a Hollywood film producer, has worked with Hallmark Films and others, is going to be back. And of course, he's a McDaniel alum as well. Please hang around afterwards. We've got cookies and punch or whatever to eat back here.
Speaking of films, though, I just want to mention that
yes, please.
November 1st, there'll be a documentary on my company on Netflix. BuzzFeed produced it, short-form documentary starting November 1st.
So mark that on your calendars, and please join me in chatting with Greg afterwards. Thanks for being here, folks.